Welcome to this lecture on reproductive biology. Today, we are going to explore one of the most fascinating and dynamic structures in the human body, the corpus luteum. We will understand how it forms, why it is absolutely essential for early pregnancy, and how its programmed regression controls the menstrual cycle. Let us dive in. To begin, let us imagine a bustling city where a massive stadium is built for a single event. Once the event is over, the city does not just abandon the stadium. Instead, they rapidly convert it into a temporary, highly efficient factory that produces essential survival supplies. In the female reproductive system, the stadium is the ovarian follicle that just released an egg. The factory it transforms into is the corpus luteum. This temporary factory works tirelessly to pump out progesterone, preparing the body for a potential pregnancy. Now that we understand the factory analogy, let us look at exactly where this structure comes from. The corpus luteum is a transient endocrine gland that forms in the ovary immediately following ovulation. When a mature Graafian follicle ruptures to release an egg, it leaves behind a shell of granulosa and theca cells. Instead of being discarded, these specific cells are repurposed to build our temporary hormone factory. This marks the very beginning of the luteal phase of the reproductive cycle. Following ovulation, these remaining cells do not just sit there. They undergo a dramatic structural and biochemical transformation known as luteinization. This entire process is driven by the luteinizing hormone secreted by the pituitary gland. Under the influence of this hormone, the cells hypertrophy, meaning they physically enlarge and change their internal machinery. Once transformed, they immediately begin synthesizing massive amounts of progesterone, alongside moderate amounts of estradiol, officially turning the structure into a fully functional endocrine gland. But what happens if this preparation is not needed? If fertilization and implantation do not occur, the corpus luteum undergoes a process called luteolysis. This is a highly programmed process of structural and functional regression. During this phase, the gland completely stops producing hormones, and its cells undergo apoptosis, which is programmed cell death. Eventually, it degrades into a harmless mass of scar tissue, known as the corpus albicans. This precise life cycle of formation, peak function, and rapid regression acts as the absolute pacemaker of the entire female reproductive cycle. Understanding this life cycle leads us to its critical biological importance. The corpus luteum is the sole biological bridge that makes early pregnancy possible. The progesterone it secretes acts like a biological glue and a rich nutrient source. It thickens the uterine lining, known as the endometrium, and keeps it highly stable so that a fertilized egg can safely implant and grow. Without this temporary hormone factory, the uterine environment would simply not be capable of supporting a new embryo during those crucial first few weeks. Because this structure is so vital, any issues with it have significant real-world consequences. Clinical problems with the corpus luteum are known as luteal phase defect. This condition is a leading cause of early miscarriages and female infertility because the uterus does not receive enough progesterone to maintain the pregnancy. Fertility specialists closely monitor these hormone levels to diagnose such issues. In fact, they often prescribe supplemental progesterone to support patients who are undergoing assisted reproductive technologies, like in vitro fertilization, ensuring the embryo has a stable environment to thrive. While reproductive biology is driven by cellular processes, We can use mathematics to understand exactly how quickly this gland shuts down. During luteolysis, the decline in progesterone is not a slow, linear drop. Instead, it follows an exponential decay curve. In this formula, P of T represents the concentration of progesterone in the blood at any given time t. P0 is the initial peak concentration, right before luteolysis begins. The constant E is the base of the natural logarithm. The variable K is the decay constant, representing how fast the cells are dying. A higher K means a faster shutdown. As time increases, the negative exponent causes the hormone levels to crash rapidly. Before we move on, let us see this formula in action with a concrete example. Imagine a patient whose corpus luteum has just begun to regress. We are given an initial peak progesterone level of 20 nanograms per milliliter and a decay constant of 0.4 per day. We want to find the concentration exactly 3 days later. We substitute these values into our exponential decay model. Multiplying the decay constant by the time gives us an exponent of negative 1.2. Evaluating this exponential term shows that only about 30% of the hormone remains. Multiplying this by the initial 20 gives a final concentration of 6.02 nanograms per milliliter. Physically, this means the hormone levels have crashed significantly in just 3 days. Now that we have calculated the concentration at a specific time, let us reverse the question to find out when menstruation will begin. Using the same patient, we know menstruation is triggered when progesterone drops to a critical threshold of 2 nanograms per milliliter. We set our target to 2 and our initial peak to 20. To solve for time, we first divide by 20 to isolate the exponential term, giving us 0.1. Next, we take the natural logarithm of both sides to bring the time variable down from the exponent. The natural log of 0.1 is approximately negative 2.302. Dividing this by the decay constant gives us exactly 5.75 days. This beautifully illustrates how the regression rate strictly dictates the biological countdown to the next menstrual cycle. To visually reinforce what we have just calculated, let us look at this graph of progesterone levels over time. On the vertical axis, we have the progesterone concentration, and on the horizontal axis, the days after ovulation. Notice the solid blue curve. It rises to a peak when the corpus luteum is fully formed and functioning. Then, as luteolysis begins, the curve drops sharply, perfectly illustrating the exponential decay we just modeled. The dashed horizontal line represents the critical menstruation threshold. Once the decaying curve crosses this line, the biological signal is sent to shed the uterine lining, starting a new cycle. We have covered a lot of ground, so let us summarize the key takeaways. First, the corpus luteum is a brilliant example of the body's adaptability, acting as a temporary hormone factory to produce progesterone. Second, this hormone is absolutely essential for preparing and maintaining the uterine lining for a potential pregnancy. Finally, if no embryo arrives, the gland undergoes luteolysis, causing an exponential drop in hormones that triggers menstruation. You now have a solid understanding of both the biological mechanisms and the mathematical modeling of the corpus luteum. Excellent work.